Right. Well, thanks, Susan and Michael, for the invitation and to the organising committee behind the scenes for the whole series. So it's a pleasure to be here today and talk about innovation and innovative technologies and their implications for diagnostics and later on for governance. And what I thought I would do, just to sort of in this opening session, is to talk about innovation at a quite broad level initially, to talk about it at a meta level, if you like, within the healthcare technology system, and set a few hairs running in respect to the sorts of features that characterise healthcare technologies today. And I've worked quite a lot, as Susan was saying, in respect to developments in emerging technologies within healthcare, whether they're in the genetics field, such as pharmacogenetics, or regenerative medicine and stem cells. And these sorts of technologies are the ones which in many ways are posing new opportunities as well as challenges for the diagnostic process, for governance and for clinicians and patients and so on. So I'm going to sketch out just a few generalities, if you like, about the healthcare system and then gradually work down to something a bit more specific. So the three parts of the brief talk, I'll talk for about 20-25 minutes or so, are related to those aspects there. The character of innovation processes today within healthcare systems, the implications that they have for innovation for diagnostics, and diagnostics and governance itself, particularly in respect to the site of governance, where we actually can find the site of governance. So let's go to the first one of these, the character of innovation processes today. So there are a number of features you could say are true of many areas of health, biotech, biomedical research. These are the sorts of features which I think disturb boundaries of medicine, disturb boundaries of disease, disturb the relationship between clinicians and patients, disturb the ways in which the site and locus of medicine itself is performed, if you like, or is placed. So you can see, if you can see, that technology is designed to be embedded, for example, the embedded nature of many healthcare technologies today through prosthetic technologies within the body of the patient. That changes that relationship between monitoring, management and control of disease and illness. Technologies are also, apart from being embedded within, are projected without the body, in the way in which, for example, in the field of, say, telemedicine, you can project pathologies across time and space and manage, monitor and scrutinise and treat indeed that whole process, through that whole process of projective nature. Technology is increasingly hybridised, bringing together different types of fields, different types of, as Martin would call it, epistemic cultures and the practices associated with them, and bringing in that way a redefinition of boundaries and disease. And you think, for example, in the field of, say, regenerative medicine, which I'm working in at the moment, you have a whole range of different types of products and therapies in that field. One of the ones which is most difficult to pull off and to get a product out of is one which is the most hybridized, and that's the so-called combination products, or combination devices. So where you have a device and a stem cell and a drug perhaps articulated together, integrated together as a form of therapy. So increasingly hybridization across disciplinary and technological boundaries. The representational power of health, particularly through imaging technologies, visualization technologies, changing the ways in which we see, literally see disease and see pathology. Think about the DNA heat maps that one sees used very regularly these days to map out the genetic pathologies associated, say, with different forms of cancer, which provide you with greater accuracy in terms of actually looking for the boundaries of disease, but at the same time produce more conundrums for the diagnostician when they can see this rather intriguing and rather complex set of arrays in the context of genomic profile. And then that leads to, in a sense, the paradox of increasing precision. So the more technologies are able to be precise in the metrics and measurement of pathology, as we move over the centuries from the stethoscope through the X-ray through to the genomic screening, that gradual shift to penetrate the body in its core leads to this fantastic power, it's empowerment in terms of precision, but in terms of prognosis and diagnosis, increasing uncertainty. The interpretive judgments of the physician, in a sense, in the early days of the stethoscope, were crucial to making sense of the symptoms. And in some ways, as you go through the genomic screening, from that early period right through to the heart of pathology of the genomes, one gets to the point where you say, well, do we need to have the interpretation of the clinician when we have that accuracy? Well, of course, it becomes even more important. So the greater the accuracy in terms of metrics, the more problematic it becomes in terms of actually what do we interpret. So contrary to the assumption that people are going to get easier to do. At the same time, and this is in many fields, not just in medicine, you would see it also at a meta level, innovation working in other ways. So one of the recent reports that some of you may have come across by the Rathenau Institute in the Netherlands, it was published, as you can see there, in 2011, Making Perfect Life, a report that was commissioned by STOA, Science Technology Options Assessment Group of the European Parliament. And the Rathenau produced this really interesting paper, which is online, you should go and see it if you haven't read it already. But they make a number of general arguments, and these are two which I think are important. Shaping healthcare, shaping the meaning of bodies, shaping the meaning of what it means to intervene. Technology becoming biology, one of their key themes that they make, and conversely, biology becoming technology. And in these different ways, they argue that interdisciplinary research within a whole range of fields, bringing together biotechnology, informatics, the new digital sciences, and so on. They argue that we're beginning to see these two types of processes at work. So technology becomes biology. You see this in the context of synthetic biology, the building, as it were, of artificial biological systems. Biology becoming technology in the context of human enhancement, sort of work that Michael Morrison has done. And we have, by the way, in the book that Susan mentioned, this book series that Susan mentioned, there will be a book coming out shortly on this question of human enhancement, the book that Susan, Sally and I edit. So those are sort of general processes at work. And then as you start to drill down in the context of the clinic, medicine, what is happening as a result of these dynamics? Well, there are two types of processes that happen. First of all, both of these processes produce a reconfiguration of where the locus of medicine actually lies, and it lies beyond the clinic. And these two particular processes are key to that. On the one hand, through the use of informatics, you can see national, international data libraries, DNA, gene biobanks, stem cell repositories, and national information systems. So these are capturing the meaning of disease and pathology, distributing that, understanding that evidence to colleagues in international networks that are monitored and governed and produced and created and reproduced by a whole range of different types of typically bioinformatic-based technologies outside the clinic in the conventional sense. And then, again related to the impact of informatics, we see new arenas within which health innovations are produced and consumed. And here I've got you can see it says Mind Medicine's Dutch website, which is deploying techniques of crowdsourcing to get patients, if you go to that site, for instance, to get patients' experiences of drugs that they're taking for chronic conditions and to some extent for acute conditions. And the Mind Medicine website is a crowdsourcing vehicle which then the organisers of that site relay, if you like, a summative set of observations from many thousands and thousands of patients in the Netherlands reporting on their use of these drugs to not only clinicians but also to drugs companies. So this is another way in which, with the formal clinical side, there's new types of information sources being deployed to shape and manage and govern innovations, how they're produced and consumed, particularly to do in this case with adverse drug reactions. And of course, crowdsourcing through social media in Web 2.0 is a really important development on a whole range of areas. Not simply in the area of health, but in areas of commercial manufacturing, in areas of reputational management, to the brand, and so on and so forth. So it's becoming an important source of evidence of some form or other, which is then being deployed in innovation systems. So these are the broad features, I think, that within the context of broad healthcare technology development are leading to a general process in which we can talk about the fragmentation and, crucially, distribution of the patient, of the body, and of healthcare data. The fragmentation and distribution. So from those first few slides, what I want to suggest is that innovation processes are leading to these types of outcomes in respect to patient, body, and data. So that will have a whole series of implications for the way in which diagnostics is undertaken, where it's undertaken, who undertakes it, why it's undertaken, and what its implications are for treatment, and so on and so forth. So those working in biomedical science, those working on the cusp between biomedical science and translation into the clinic, are confronting these types of opportunities and these challenges and these new technologies on a regular basis and having to make sense of them in their everyday practice. So what we see then with all these processes is, as this slide says, the interplay of the social and the technical and the social. And one area you can see that in particular is in the field that I've been working on with Michael when he was in York, especially the field of regenerative medicine or tissue engineering more generally. And this is a classic site where you see the social and the technical come together. Tissue engineering itself is a difficult task. It's been around for many years, a difficult type of technology. Engineering tissue to work with polymers, for example, to produce a scaffold, a tissue-engineered scaffold, which then would be introduced into the body as a form of device, as a form of treatment for a particular type of lesion or a particular type of replacement of bone, or what it may be. Now that process of tissue engineering and building this scaffold is paralleled at the same time by a more social process, if you like a social scaffold. Putting in place all the type of governance arrangements, putting in place all the ethical arrangements, putting in place all the investment arrangements, putting in place all the ways in which the state legislates this particular field of inquiry and development. So in many ways, tissue engineering has to be accompanied by, if you like, and I don't mean this in the Darwinian, social Darwinian sense, a form of social engineering, a form of social management process as well, a social scaffold at the same time. So modern technologies are, and clearly this has been true for many, many centuries, but in particular you see this here, that these technologies do require a substantial amount of social investment to make them work, a form of sort of social engineering, as I say. So the very fibres of the scaffold that's implanted in a body, the very structure of a tissue-engineered scaffold, carry with it the imprint, the embodiment of this social engineering, the social scaffold at the same time. So when we're thinking about areas like stem cells, and you think about a stem cell being implanted in the body, whether it's autologous or allogeneic, that implantation process carries behind it the weight of a social process that's made that possible, And of course, different types of processes carry different types of social implications. So, what are the implications of these innovation processes for diagnostic technologies? One of the ways in which I've been recently exploring this in York is through a study that was completed recently with a colleague of mine, Helen Cox, and it was in connection with the screening of patients for hematological malignancy. And so these are particularly patients who might suffer from leukemia. And in the study, which was published not too long ago in Health, in I think it was October of 2012, sorry, you can see in that report that Helen Cox and I did on this project, the ways in which diagnostics works in this particular setting becomes more and more difficult to manage as a result of the increasing predictive power and metrics of hematological malignancy diagnostic systems, particularly using molecular tests. So these molecular genetic tests that are used now for diagnosing these pathologies, diagnosing these disorders, are increasingly vanishingly small in terms of their levels of precision, levels of accuracy. And they produce disorders that don't have any clinical symptoms. So these patients may or may not actually go down with leukemia, ever. And so what's happened in the context of understanding the use of this particular type of information is that the clinicians, the biomedical researchers, those working in the labs, working closely with these, have had to produce new types of labeling for these different types of condition. So, for example, the situation where things are clinically silent and there is no symptomatology at all, even though there's an apparent predisposition towards a particular disorder, but now, rather than talking about leukemia or the possibilities of leukemia, they're now talking about this preclinical lymphocytic leukemia as, quote, monoclonal B-cell lymphocytosis. And that's addressed the issue of informing patients that they have an abnormality in their blood without the use of the leukemia label, with all the associated connotations of such a label. No formal requirement to determine risk as such. And similarly, preclinical myeloma has been relabeled, quote, monoclonal thermopathy of undetermined significance in the US. So those sorts of shifts in the way, or these attempts to manage this type of subclinical, clinically silent, occult diseases, as they're called, is indicative of the way in which this paradox I talked about before is apparent in many particular areas of contemporary biomedical science. Now, what are then the implications of all this for diagnostics and governance? And I want to particularly talk about the forms of governance and the science of governance as a result of these significant shifts. First of all, when we're thinking about the oversight of the impact of these highly sophisticated advanced technologies, whether they are a form of molecular diagnostic testing, in the field of hematology, or tissue engineering, or bioinformatics and so on, we have to think about that oversight only working in a particular context, understanding the context itself. So what is the context in which diagnostics is deployed? Well, I've got 3 points here I want to make about the context in which diagnostics is deployed, which have implications for governance itself. First of all, if you think about innovation and the Scrabble metaphor that Michel Fallon very astutely provided for us some years ago. And he talks about this. He says, a market is like a Scrabble board. There is no point in wanting to place an innovation that does not correspond to the possibilities it affords. There's no point in keeping aside the perfect word. So if you play Scrabble and you've got that 7-letter word and you want to put it down, but there's no one on the board you can put it down because all the board's been full up, there's no point holding on to it. You have to go with the flow, as it were, go with the letters and break that up and put it into 2 words, whatever it may be, and get 3 instead of 103. And innovation is like that. Innovation works in the context, that is to say, innovation does not come, as it were, pre-packaged and then is deployed. It does what it says on the lid. It doesn't. Different types of innovation work in different ways. The same type of innovation will work in different ways, in different contexts. This is particularly true of diagnostic technologies. So if you have a diagnostic technology, for example, a biomarker, or some of the specialist developments in regenerative medicine, such as nanolite technologies, that are to act as an early screening device for certain types of disease, such as surgery with HIV and AIDS. That type of technology will only make sense. It's like you having to change the letters, as it were, on a Scrabble board, as you go from a particular type of clinic, a hospital, to a long-term chronic care clinic, to a different type of set of arrangements, which might be care in the community. So as you go from one organisational to another organisational to another organisational setting, institutional setting, this particular device will have more or less utility from A to B to C. So the same thing actually is performed in different ways and has more or less workability. Different types of time frames, different types of spatial dynamics to it, different types of workability. So how you introduce that innovation has to map onto what spare places on the board of the clinical practice board are available to put your Scrabble letters down, to put your innovation device down. So that means that just as in Scrabble, you've got to, and the governance of Scrabble is, hey, I'm not going to accept that word, let's consult the dictionary. Is it there really in the dictionary? Or that's actually a real name from whatever, Sweden. So you can't have that. So there's a similar type of debate that goes on in these settings about the workability and the utility and the governability in terms of resource, in terms of responsibility, in terms of decision making, in terms of control, in terms of liability over these devices as they go from A to B to C. Very, very different type of take-up. So context, so if these are diagnostic devices and those contexts are significant, context can be in some ways too strong. You know, those working in STS say context is everything. So that was a good thing to understand, yes. But if you want to talk about it in terms of translational medicine, you could say from an STS point of view, context sometimes is too strong to make that innovation work. The other point about it is that innovation is distributed, it's non-linear. The model of innovation has to be understood in those terms. It's recursive, it's non-linear, it's distributed across a range of different types of social actors. And therefore governance, in that sense, becomes distributable across these different innovation settings. And then finally, the third point I want to make about context, from work that many of you will know, Carl May, down in Southampton, with his theory of normalization process theory and his ideas about workability. It's an excellent work. If you go to the website, you'll see he has an e-toolkit for normalization, which is deployed and used now in the NHS and Department of Health and other agencies internationally. And it's all about this question to do with workability. How are things normalized? So when you talk about diagnostic tools, what Carl's work shows is that there's a whole series of organizational, skills-based, epistemic-based processes and factors at work that shape the utility of particular types of device, intervention, therapies, and so on. So diagnostics, again, as device intervention therapy, device intervention, in the sense of making sense of pathology, have to be normalized. They can be adopted, but they have to be also then subsequently normalized in different ways. So these are three broad contextual related issues which shape the uptake, the sense-making, the performability of and performance of a particular type of diagnostic. And therefore, the ways in which, from a governance point of view, these are overseen. So these are three processes at work, which I think that relate to governance regimes associated with diagnostic tests per se. First of all, the first point is to say that technology has so far been centralizing, in the sense of gathering in information, controlling. From a sort of regime point of view, technology has enabled and empowered powerful social actors, medics, for example, in this context, to be the authority and the source and locus of wisdom, treatment, and intervention, and governance. As technology then becomes more distributed in the ways I described earlier on in this brief presentation, that centralizing process gradually starts to unpick and becomes a much more open process. So how does, if it becomes more distributive, how then, what implications might have that for regulation, for safety, for clinical governance? Where does the site of governance lie? Take that crowdsourcing example. How is governance work in the context of the Mind Medicine website in the Netherlands with regard to responsibility, liability, accountability? There's no legislation that covers that. Whose voice is being heard, and how is that voice being given some sort of diagnostic power? And this process also, in terms of innovation, tends to involve a deinstitutionalization of governance, and so subvert the notion of institutional rules of coordination. A very important point, that often governance is defined as a form of coordination which is institutionalized. institutional rules of coordination. That sort of notion becomes subject to challenge and disruption as these processes deinstitutionalize. Again, crowdsourcing is a deinstitutionalization of coordination. And there's no isomorphic relation between a particular technology introduced and a specific regime of governance overseeing it. So in other words, technology A, you cannot read off from that it would generate a governance regime that will be true and commonly found as you move from country A to country B to country C to country D. So if you take the field of regenerative medicine and stem cell science, that technology, as it moves from country A to country C, has a different type of governance regime overseeing it, is defined in different ways, and performs in different sorts of ways too. So there's no isomorphic relationship. You can't read off in that particular way. So to conclude, when we're thinking about innovation and its relationship to diagnostics and governance, looking at this very broad level, I've just been talking about this at a very general level, we need to think about innovation in terms of the way it reorders and perhaps disorders patient-clinician relationships, in that sense. So it reorders them and it disorders them. It reorders them and has new forms of control, oversight, disorders them by breaking things up in ways which might create new problems for those involved, for patients and clinicians. And secondly, we have to ask what impact has this for the oversight of diagnostics and the locus of responsibility. The locus of responsibility is out with the clinical, the doctor's black bag as a result of these types of processes. And so we have a whole range of agencies, regulatory agencies, such as Chris working NICE, that try to provide that type of oversight and governance out with the clinic and provide feedback into clinical context. And it's a difficult task and a challenge, made increasingly difficult as a result of the way in which this reordering and disordering of patient-clinical relationships goes on. So those are a few general observations, I think, based upon my experience of working in this field of health innovation for many years, where I try to sketch out some general processes at work at a meta level, and then try to say how they have implications for specifics to do with diagnostics, such as in the field of hematology and malignancy, and making sense of the increasing powers of metrics and metricization of disease through molecular genetic testing, and the increasing uncertainties that creates, and how that then has to be resolved, the new types of labeling associated with those pathologies, which may or may not actually be clinically ever come out in terms of patient illnesses. And then thirdly, how that then impacts on governance. So governance is, yes, about rules of coordination, but the rules of coordination are highly contextualized. They're about ways in which we can bring together and some sort of symmetry between, on the one hand, workability of innovation, and on the other hand, accountability of innovation. Those two things, workability and accountability, are crucial to the relation between governance and innovation. And on that point, I shall stop. Thanks.